Welcome to the 8th Annual Seattle Area.
The Seattle Epidemiologic Research and Information Center, in collaboration with the Department of Veterans Affairs, the VA Employee Education System, and the University of Washington Departments of Epidemiology and Biostatistics, presents the 8th Annual Epidemiology, Biostatistics, and Clinical Research Methods Summer Session.
Welcome to the 8th Annual Seattle ERIC Epidemiology, Biostatistics, and Clinical Research Methods Summer Session. My name is Ed Boyko. I'm the course director. The title of today's course is Genetic Epidemiology. I'm pleased to introduce to you today our speaker, Dr. Karen Edwards, an associate professor in the Department of Epidemiology and the Institute for Public Health Genetics. She's also director of the Center for Genomics and Public Health at the University of Washington. The title of her talk today is Linkage Analysis and Pedigree Checking. Dr. Edwards.
Good morning, everybody. Today we're talking about linkage analyses and also pedigree checking. So before we actually jump into talking about linkage analysis, I wanted to spend a few minutes reminding everybody about how important it is to actually check the structure of your pedigrees. You can assume at this point, if you have family data, that you've probably spent a fair amount of time collecting this data. And before you actually jump in and start doing linkage analysis, it is critical that you actually check your pedigrees and your structure to make sure that they're accurate. At this point, you should have genotypic data available. And so now what you'll do before you actually begin a linkage analysis is use your genotypic data to make sure that your individuals in your pedigrees are related the way that you have them indicated in your database. A couple of the things that you look for right up front is making sure that your genders are correct. Okay? Everybody in your pedigree has to have a mother and a father, and we're talking about biological relationships now. So you have to make sure that you've got both genders represented in the parents. If the genders are incorrectly displayed, you'll run into problems, and that's not uncommon to find data entry errors around gender. Also, checking the structure of your pedigree. As I indicated, we have to make sure at this point that people are related the way that you have them displayed. And one of the most common structural errors in a pedigree is that there may be one child who does not share the same father as the other children in that nuclear family. Okay? This isn't necessarily a structural error, but from an analysis standpoint, this will cause problems. Okay? So you need to make sure that all the relationships are correct. What we tend to do is to use the genetic markers that we have genotyped to determine whether or not our individuals share marker alleles as we would expect given the biological relationships that are noted on the pedigrees. So for example, what we'd be doing is using our genetic markers to make sure that parents share 50% of their genetic material with the offspring. Okay? If a parent at a particular locus has the allele A, we would look to see then in the offspring generation, do the children share the same alleles as their parents? And I'll draw out some examples in a few minutes. But these are critical things to check, and you need to check them with more than one marker. Okay? Usually we take some percentage of our markers, run a first pass through the pedigrees, check for huge obvious errors, and then we go through and actually run all of the markers that we have available to us to make sure that they are inherited in the way that we think they should be. What we tend to do is find the big errors first, for example, these situations where an offspring may not be related to the pedigree as we think they should be, and we exclude those individuals right off the bat. But you'll find with individual markers, sometimes they don't run, the analyses, the laboratory analyses don't work properly, and you may find that you have problems with individual markers that are consistent throughout many, many different pedigrees. And so those markers you may actually want to exclude from your analyses as well. So we're kind of checking at two levels here. First of all, whether our pedigree structures are correct, and then secondly, whether or not the actual markers are correct, or if there looks like there's problems in the genotyping of particular markers. And it's not uncommon for some of these to not run very well. Okay? We also said that one of the important things when you're thinking about using family data to do genetic linkage analyses in particular is that you have to have files that indicate how people are related to each other. And the way that you accomplish that is to, for every individual in your pedigree who's related to somebody else, you have to have parental information so that everybody is linked up that way. Now, I'll draw an example here in a second when we draw a pedigree and show you what I'm talking about. But for example, if you're studying a late onset disease, and let's say that your case, your proband who brought the family to attention, let's say is 65 years old. They have 5 living siblings who are also included in your study. Obviously, well, not obviously, but very likely both parents are deceased from that generation. So you've got a sibship with 5 offspring. Parents are deceased. The only way that you can correctly include those people in your data file is to include what we call dummy variables or dummy parents. Even though they're deceased, you have to put them in because that's what links those 5 siblings together. So without that parental information in your structure file, those people will not be linked with each other. So there's a lot of data manipulation and adding of individuals to the pedigrees to make these files complete, and it takes a long time to do this. The other situation that we encounter quite often is if you're working in a family, let's say you've got those 5 siblings, you collect their children, and in some cases those people also have grandchildren. What we see quite often is the spouse of the child, so the second parent of the grandchildren, says, well, I'm not biologically related to the case, therefore my participation is not important. Well, if you're going to include those grandchildren, you actually do need the spouse's information. And so sometimes it's a little challenging to convince the spouses that they also need to participate in the study or that their information is valuable. Okay? But if they don't participate, you still have to include a dummy variable for that other parent on your pedigree. Otherwise, those children will not be linked together and will not show that they're related in your structure file. So as I said, there's a lot of adding of individuals to your pedigrees once you've collected your family data. And I'll draw it out in a second so that makes more sense to you. The other thing is that most of the linkage analyses and most of these software packages assume that when you have 2 people in the same sibship that they're going to be siblings. If you have MZ twins, you need to exclude them from the analysis. Okay? Now frequently, rather than excluding both twins, you randomly select one and keep one in and take one out. Because remember, MZ twins are genetically identical and they're not independent observations from that standpoint. Okay, so you need to take one out. If not, you'll get errors in your analyses that say something about the inability to successfully compute IBDs and other things. So you have to take at least one of the MZ co-twins out. Now, this idea of checking for inconsistency sounds pretty straightforward and pretty simple, but it's one of the most time-consuming things that we do in linkage analyses, and particularly as we increase the number of markers that we're using in these studies. Traditionally, linkage analyses that we're going to talk about today use what we call microsatellite markers, and generally what we have is about 380 microsatellite markers that span the genome that we use for our linkage analyses. So you can imagine that what you're having to do is go through your pedigrees using all 380 of those markers, checking to make sure that each one is consistent within your pedigrees. This is an iterative process. These programs, they look for big problems first, and when they encounter a problem, they just stop running. You have to go in, fix that problem before it will continue running through the pedigrees. And so you have to do this over and over and over again. And sometimes it is very difficult to identify what the exact error is, and what you may end up having to do is exclude a whole portion of the pedigree because you can't determine definitively who the problem individual is. So this is one of the issues in dealing with humans rather than animals that presents quite a few challenges to us, is getting these structures correct and getting the biological relationships to be accurate. Okay, in terms of software that's available for checking for these inconsistencies, there are several packages out there. Most of them, the documentation is pretty good. It's like a lot of these things. The first time you run it, it's going to take you a while to figure out how to get things moving and how to interpret the output. But once you're used to it, most people stick with a package because they know how to make it run and they understand what it's doing. So you'll find that there's a cohort effect in terms of what kind of software people use, that they keep using it because you know what's going on. A couple of the ones that are most commonly seen out there is a software package called PedCheck, which is available free off the web. It's quite a good package. Does fairly well at detecting the major sources of structural errors in the pedigrees. Another software package, which I haven't mentioned before, is called, oh, it's not, well, there's Mendel and there's Merlin. There are a couple of different software packages out there. And then Sage, I keep mentioning again because this is one that I've mentioned a couple of times that will do a variety of different tasks in genetic epidemiology. The other thing that's quite helpful to do before you really sit down and start doing an analysis, a linkage analysis or any kind of analysis that involves your family data, is to draw out your pedigrees. This is, again, something that can be time consuming, and it's a good project for a graduate student if you have any, but to have them sit down and actually draw out the pedigrees so you can take a look at the structure of the pedigrees as you have it in your database. As you do your pedigree checking, you can refer back to the pedigrees, and it just makes it a lot easier. There frequently are a lot of decisions you have to make about data. If you have to break up pedigrees, you need to be able to look at them and make decisions about things. And just, it helps you to visualize your data. Okay? We're moving into a new area now where you can't just do a histogram. You've got related individuals, you have a lot of data, and drawing the pedigrees is extremely helpful. Again, there are a number of software packages out there that are available. Cyrillic and Progeny are two common ones. Cyrillic is not free anymore. Progeny, I don't remember the cost. The other thing that is helping us in many ways is a lot of people are interested in genealogy these days. And there actually are a lot of packages out there on the web that will help you to draw your pedigrees. They're available free. They may not do some of the fancy things that the programs that are designed specifically for genetic epidemiologic analyses will do, but they're frequently very easy to use. And so if you just want something free to draw pedigrees with, you can search on the web under a lot of these genealogy websites, and there is a lot of software out there. Okay. The other thing to consider before you even begin your analysis is how you're going to handle this data. And in particular, how are you going to keep track of these inconsistencies that you've identified in the pedigree checking phase? And let me give you two different types of inconsistencies that you need to consider. The first is what we call the Mendelian inconsistencies. So for example, non-paternities might fit under that category. If you identify a sibling within a sibship that is inconsistent with the rest of the siblings in that pedigree and you attribute it to either non-paternity or laboratory error, we shouldn't make judgments here, you have to decide what you're going to do with that sibling. You have a couple of options. Because it's not related to the rest of the siblings as a full biologic sibling, you need to either correct the relationship and make it a half-sib. If you made it a half-sibling, what would you have to do to your data file to indicate that that's a half-sib?
Insert the father.
Exactly. So you'd have to insert another father for that sibling. That's exactly right. Okay, so you can do that. That's one option. The other option is you exclude that individual from your pedigree. Okay, we hate to throw away data. So frequently what we'll do is insert another biological father and make that child a half-sib with the rest. Okay, so if you, yes? Or mother. Yep, that's correct. We generally don't run into non-maternities very often, but it is possible. And frequently what you get is when people are reporting the relationships, they don't report that it's a stepmother or a stepfather. And so this is why I said we don't necessarily assume it's a non-paternity per se, but in terms of the biological relationships, that child is not related to the rest by either the father or the mother. Okay? And so you've got to correct that error and add in the appropriate other relative. Okay? So typically what we do is we add in the other spouse and make it a half-sibling. But sometimes there may be situations where you want to not include that child in the pedigree as it is because it doesn't appear to be related to either the father or the mother. And in those cases, we have no idea what's going on. And so those individuals, if you wanted to, you would make two new parents, but then they're not related to the rest of the pedigree. So in that situation, it's probably better to exclude the data for that individual from your analyses. Now the question comes in, how do you do that? Do you delete the data from your database and just strike that person from your data files? Well, like I said, we just hate to throw away data. So the approach that we've taken in a couple of other groups is we create a new indicator variable that we name something like Mendel to indicate whether or not each individual in the pedigree is Mendelian consistent or inconsistent. And then when we actually get ready to do the analyses, we say exclude everybody who Mendel equals 1, which would be, inconsistent or however you code it, so that that individual is actually in our file but doesn't go into any linkage analyses or any analyses that rely on family structure data. The reason we don't throw them out is there are often times where you want to go back and look at unrelated subjects to get estimates of allele frequencies or to do other analyses with. If you wanted to select out a sample of unrelated subjects to do a case control study, that person then could actually be valuable. Okay? So we don't actually delete them from our database. Now the other situation you're going to have, so for subjects who we've identified as being some problem with their genotypes, whether it just does not match anybody in the pedigree, but what you see with these individuals is, let's say you genotype 380 markers. You're going to find that individuals who are problematic in terms of the structure of the pedigree have errors on the majority of these markers. Now by chance, some of them will work fine, and for some markers that are not very informative, they may not indicate that there's a problem with the structure of the file. So it's not that they're going to be inconsistent at every single marker, but it will be for the vast majority of them. Probably over at least 50% of the markers will come up as being problematic. Okay? So those people, clearly, we would indicate those as non-Mendelian and exclude them from the analyses. But then you have situations where individuals, there may be, let's say, between 20 markers and 50 markers that are problematic for an individual. Is that enough to indicate that that subject should be excluded from your pedigree? Probably not. That may be just an issue with those 20 to 50 markers having problems for that particular individual. So in those situations, what we do is we blank the data for those individual markers. Now you can see how time-consuming this is to do this for every subject and every marker. So there, as I said, are 2 levels of data checking, and that's why this takes such a long time, that you frequently have to go through and make decisions about who it is you're excluding, what markers you're excluding, how you're going to deal with this data. But as I said, if you don't take the time at this step, you're going to make it more difficult for yourselves to actually detect linkage. Okay? Okay, so now let's say that you've spent your year after collecting your family data, cleaning it and checking it, getting your files ready to run for linkage analysis. This is kind of the premier sort of analyses that people want to get to immediately, is the linkage analysis. Most of the things we've been talking about up to this point are really to help you think about the phenotype, but this is sort of the holy grail of genetic epidemiology, is linkage analysis and genetic association studies. Because really the goal for most individuals working in this area is to identify the gene or genes that increase risk for a particular disease or a particular trait. And linkage analysis is one of the workhorses of that goal. So the aim of linkage analysis, and this is going to sound a little odd at first, but we'll talk about it, is to infer the relative positions of 2 or more loci. The idea here is that we have genetic markers, and I'll just define what a genetic marker is, whose location on the chromosome is known. So you can think about these markers as signposts along the chromosome. You know where they're located. They may not have any function that you know of, but you know their position on the chromosome. And what you're doing is using those markers whose positions are known to help you localize where the susceptibility genes are. And these susceptibility genes are what we're looking for, but whose location we have no idea where it is. We assume that they exist based on all the preliminary work we've done to this point. We've shown that the trait is heritable. We've shown all the things that we've talked about in here. So we know that it's very likely that a gene exists for that trait. Now our goal is to say, all right, where is it? And you can think of this in broad terms, and especially with linkage analysis, is that this is a pretty crude mapping approach. It's not quite as crude as what I'm going to say, but it's It's not localizing us to a particular exact location, but it's putting us somewhere on a chromosome, the region which may be quite large and contain a large number of candidate genes, but it's at least saying, okay, on this chromosome, on this arm, in this region, it appears that there's a candidate gene or a gene that's influencing your trait of interest. Okay? So that's what we're doing with linkage analysis. We're using the location of known markers to help us localize where our unknown susceptibility gene is. Okay? And we really take this as sort of the ultimate statistical evidence that a gene exists. Keep in mind with linkage analysis, in some situations, we still don't know what the gene is, but we at least have stronger evidence that it actually exists because now we've identified a chromosomal location that appears to co-segregate with our susceptibility gene. Okay, so the basic idea behind linkage analysis is kind of what it sounds like. The name implies that these two loci are linked together and co-segregate in the families. So the basic concept here is that the closer together these two things are, the stronger the correlation or the co-segregation will be. The farther apart they are, the more often we will get recombination events which breaks the relationship between these two markers. Now there's some terms I'm going to have to define, but let's go with this for a while. Okay? So the idea is if we have two loci, okay, two genes that are very close together, that they will segregate within the family as a unit. Okay? And that's really what we're relying on in linkage analysis, is to look for the co-segregation of these things through the pedigree. Whenever we see this relationship being broken, we say, oh, okay, so it's a little bit farther apart than we think. We use this concept of recombination to estimate how far apart these things actually are. Okay? In general, the markers that we use, especially for the traditional LOD-based linkage analysis where we use a microsatellite scan, these markers are on average about 10 centimorgans apart, which that may not mean anything to you, but they're not right next to each other. Okay, that's a fairly big distance that can contain a large number of genes. The idea that you're hoping for is that between each of these markers, your gene of interest, your susceptibility gene, is going to be somewhere in between there. And the closer it is to one of those particular markers, the stronger your evidence for linkage will be. Okay? So this is why when we start talking about SNPs, people are very excited about the use of SNPs because they're much more frequent than microsatellite markers and you can place them more finely across the genome, with the idea being that if you've got a finer spacing between these markers, you're much more likely to be very close to your susceptibility gene. So that's the concept behind SNPs. But we'll talk first about microsatellite markers and then about SNPs. But the whole idea with these genetic markers is that they're marking locations on the chromosomes, the idea being that you hope your susceptibility gene is close to one of those things. Okay? That's the whole concept behind linkage analysis. It's obviously a simplification, but it's not inaccurate to simplify it to that level. So the other thing that's important, and this will make more sense when we start talking about association studies, is when we're talking about linkage, what we're looking for is at the genetic marker, we have different alleles, which we've talked about, and what you're looking for is, does a particular allele, do alleles at that locus co-segregate with your gene of interest? In linkage analysis, you actually do not care what that allele is. Okay? You're just using those alleles at that locus as markers to see if they co-segregate with your disease. Because you can imagine if we have a locus and it has a number of possible alleles, let's say you've got 10 different alleles that you could see at that location. That's what we call a polymorphic marker. Microsatellite markers, tandem repeats, things like that tend to be very polymorphic, and there are generally more than 2 forms of that particular marker, or of the alleles at that particular location. And so if you think about it, in different families, based on what the parental genotypes are, you could see different alleles at that marker locus segregating in the family. So for linkage analysis, we actually don't care what allele is segregating. We're just using it, as I said, as a marker to the location. When we move to association studies, however, it's a whole different ballgame. There, what we're actually saying is a particular allele associated with our disease of interest. And it's a very different concept. Okay? So for right now, we don't care what allele segregates. We're just using that to say, do the alleles, whatever they are in a particular family, co-segregate with our disease. We don't care if there are different alleles in different families, and it's very likely that there will be. Does that make sense to everybody? Okay. So, as I said, because we're using traditionally in linkage analysis the set of microsatellite markers, they tend to be very polymorphic, and there's a reason for that, and I'll show you in a second. So that it's, as I said, not uncommon to see different alleles at each marker present in different families. Some of these markers are very polymorphic and can have 20 different alleles at that particular location. Okay, so as I said, kind of the first step in this process is to do your linkage analyses, and hopefully what you end up with at the end is some region on a chromosome that shows evidence for linkage. This is only the beginning of the journey to identify what that gene is. And as genetic epidemiologists, our job in many cases stops there. Now we may lay down additional markers and try to refine the map a little bit, but this actually involves the actual identification of the gene involves many more people than just the genetic epidemiologist. Once you've identified a region and narrowed it down so that it's small enough, you may have molecular biologists come in, sequence the region, identify what's in there. Once an actual candidate functional variant has been identified, then frequently you need to understand what the function of that variant is, how it affects disease, and so on and so forth. So the localization to a particular point on the chromosome is really just the beginning of trying to figure out what gene is in there, what it does, and how it functions. And so in genetic epi these days, it's pretty important to have collaborators who do that type of work with you, because otherwise you're only going to get so far, and it's not very satisfying to say, well, we've got a region that's 20 centimorgans long and there's, 25 different genes in here, but we don't know which one it is. It's like getting close but not quite getting to the end. So it's important to think about having teams that can help you get a little further along. Yes?
So once you've identified a region through a linkage analysis, does it make sense to go in and identify a part of that region and do another linkage analysis? And if that's the case, then when does this all end? When you're at a single gene level?
Yeah, that's a really good question. And there are a variety of different approaches that people are using to kind of narrow the region. And this is what we refer to as fine mapping. Okay? In some cases, what you may want to do is take your families, lay down more SNPs or more markers in the region, and narrow that region down so that you've, if you go to a molecular biologist, is anybody a molecular biologist in here? Okay, darn. If you go to them with this big region, they'll say, we can't possibly sequence this. So you've got to narrow it down to something that's manageable that they might be able to sequence. And so that's where the fine mapping comes in. Now, some people will take that approach where they just take a region, lay down more markers, try to narrow the region. What other people may do is look in that region and say, well, what candidate genes are in here? And then they may say, well, there are 25 genes in here, but only 3 of them seem like they're going to be good candidates for our disease. So they may look specifically at those candidate genes. That's a little risky because for the most part, we just don't understand that much about the function and the biology of these genes to really pick them with any, I won't say with any confidence. We frequently pick candidate genes because we think, based on what we know, that they're going to be the gene, and frequently they turn out not to be. We identify other genes who we never would have thought of as being involved. So there really are 2 approaches here. I would say most people kind of do a little of each. They lay down more markers in the region. If there are candidate genes in that region, they make sure that the candidate genes are covered very thoroughly so that if they're involved, you're going to pick it up. Now, some people will continue to use the families, do another linkage analysis or a family-based test of association. Other people at that point will then pick out a case control sample and do a case control association study. Okay, so you have a couple of options here, and it kind of depends on what data you have available to you and how you proceed. But this is where it gets really interesting is, now you've got this region, what are you going to do? Yes?
Are there any genetic epidemiologists who either do their own sequencing or on their teams have people who can sequence?
Yeah. There aren't too many of us that do that, just because it's very time-consuming to kind of be in the lab and do both. But most of us have collaborators that's what they do, and they do well. And so it's, you have to have a lab that's geared up and able to do that because, most of these things, it's pretty competitive. You're not the only one out there looking for these genes. And so in many cases, if you find something, you've got to be able to go in there fairly quickly and figure out what's going on. Or you end up replicating somebody else's result. And that's okay, too. But it's, people like to be the first. Okay. So as I said, the whole goal with these linkage analyses is first to identify that region that even harbors your susceptibility genes. And there are generally two approaches that people take to do that. Okay. The first is what we call the genome-wide scan. And what we're doing with this approach is we're saying, we have pretty good evidence from all of our preliminary work that at least one gene exists somewhere in the genome. Rather than take a chance and try to pick out some candidate genes, let's just look across the genome and see if we can identify a region that appears to harbor our susceptibility gene. So what we're doing here is trying to identify that susceptibility gene based purely on location. Okay? And so this is what I was describing a second ago, where we use these genetic markers to try and pinpoint a region of the genome where our susceptibility gene lies. Okay? So we're not picking regions based on what genes are in there or what function is in there. We're just saying, let's look across the genome without regard to function, see if we can identify a region. Sometimes people refer to this as the positional genetic approach. It's a genome-wide scan, usually, okay, where you've got markers evenly spaced across the genome. Up until recently, these approaches, we used microsatellite markers, as I said, about 380 microsatellite markers spanning the genome with an average spacing of about 10 centimorgans. Now it's become increasingly possible to use very dense SNP maps to do this. We could spend a lot of time talking about the pros and cons of SNPs versus microsatellites, especially in the family-based studies. But the basic concept with the SNP maps is that, as I said, there are many, many, many more SNPs in the genome. The problem with the SNPs is they're not polymorphic, they're biallelic. So there's only two possible alleles at each of these SNPs. So they're not very informative individually. But where they give you the greater power is that there are so many of them across the genome. And there now are SNPs, there are several companies who make genotyping approaches where you can genotype up to 500,000 SNPs. So we're going from 380 microsatellite markers to 500,000 SNPs. Computationally, there are some challenges in using the SNP data with family pedigrees. These things take a long time to run. We spent 2 years ago a whole summer running some of these SNP scans, and it is really computationally intensive. We had every computer in our group running these analyses, and we ran for like 2 months solid just to do the basic IBD calculations. It's quite intensive, and it's a lot of data to deal with. And then the issues of pedigree checking become even more complex when you've got that much data. So while there are some advantages, this isn't something you can just jump into and do like you would with the microsatellite scan. So with the family-based studies, it's these data are out there. You just have to think about how to use them. Yeah. My
understanding is that if you contract out to a company like Affymetrix or whatever, it's about down to $1,000 per subject. So if you have a large number of subjects, that can be really expensive. Yeah. But there's been a real improvement in cost. Is that? That's true. Yeah. Yeah. It's it was not feasible for any single investigator to do this as part of an R01 because these things were so expensive. Even now, at $1,000 a subject, it's still, if you've got a big set of families, you're not going to be under that $500,000 direct cost rate
to do this type of genotyping. Yeah.
I just want to make sure I understand the difference between a SNP and an allele.
Okay.
So a SNP is a base pair, so it's either got to be adenine or guanine or cytosine or thymine, depending on what the other one is. So how many SNPs go into an allele?
Right. So a SNP, yeah, I knew I needed to define these markers. So let's start with the microsatellites. So the microsatellites at a particular location, they can have many different alleles present, so many different variants of that particular gene. With a SNP again is a location, and what you have at that particular location is one or two forms of the variant based on what the underlying base pairs are. So what we're saying is that with these SNPs, they're biallelic. There's only two possible forms of the SNP. What's nice about them is that there are so many of them that you can have a SNP every couple of centimorgans if you do these very dense SNP scans. But because there's only two alleles, they're not very informative. And when I talk about informativeness, you'll see when we talk about identity by descent estimation, what you need to be able to track in these pedigrees is what allele you got from your father, which one you got from your mother, and you need to be able to trace that through the pedigree. If there are only two alleles, by chance, half your subjects, half your family members could have one form of the allele, and the others could have the other form. And so being able to track which allele came from which parent through the pedigree is much more difficult than if you have 20 different markers that you can trace pretty carefully what came from where. So this is when people talk about SNPs, they say the advantages are there are many more of them, so they're closer together. The disadvantage is that each one individually is not very informative, but together, if you look at haplotypes, it's more informative. Does that answer the question?
Yeah. Partly, before there were SNPs, there were alleles. And so how was so allele meant something different in the past. And I'm wondering, I mean, in Mendel's time, it was, I don't know, blue eyes, brown eyes, but so it seems to have different meanings over time.
That's true. It hasn't, let me see if I can give an example that would make this work. So let's take eye color. So let's say that the big B allele gave you brown eyes, the little b gave you blue eyes, and it was recessive. So you had to have two copies of the little b allele to get blue eyes. So the big B and the little b are alleles. That's what we refer to as an allele. Now, if that was not a gene, let's say it was a SNP, you could still have the big B and the little b, and those are still called alleles. Okay, but they're the alleles at that location in the SNP. So it's still the same concept. People just refer to them slightly differently. An allele is just a variant of the gene. It's an alternative configuration, if you will, but it's at a location. So keep in mind with these genes that there are many, many base pairs, and there are many opportunities within a gene for there to be variations. Okay? Within these genes, we have SNPs, we have microsatellite markers, we have other sorts of things that we use to say, where in the gene are we? To mark a location, a locus, physical location in that gene so that we can, as I said, try to place our unknown gene next to something that we know its position. Okay, so that's, when we talk about allele, we just mean the alternative configurations of a particular location. There's a lot of going back and forth with terminology in genetic epidemiology. People use a lot of these things interchangeably, which does not make it easy to follow. I try to be good, but I slip into that as well. Okay. So the one approach with family data, as I said, is this genome-wide scan approach. And that, as I said, we're not using any hypotheses going into the linkage scan other than there is a gene that exists for our disease. The other approach, which was pretty popular for a while as well, is the candidate gene approach. And that's where, based on the underlying biology, you have identified genes who we know about, so genes who have been identified, maybe whose function is understood somewhat, whose structure is known, that you say, this gene influences cholesterol metabolism. Therefore, I think this is a good candidate gene for cardiovascular disease. for example. And so you may go through and pick out candidate genes whose, based on the biology or some function, you think is involved in your disease. That's a candidate gene approach. And in that situation, what you do is you identify markers within your candidate gene, whether they be microsatellites or SNPs, and then you test for linkage with those markers only in that gene or around that gene. Okay? So that's different from the genome-wide scan approach. Here you're saying, we're actually looking at particular candidates because we think they're involved in the disease. Sometimes that's turned out well. For the most part, the candidate gene studies have not been that successful. And we're not quite sure why that is. Many times things can't be replicated. We don't know if the first report was a false positive, if there were different situations in different populations, or what, or if we didn't have enough power. But it's been a little difficult to replicate some of these, and I think people have kind of shied away a little bit from the candidate gene approach and gone back to these more genome-wide approaches using the denser SNPs that we now have available. But people still do it. And as a second stage, you might want to consider a candidate gene approach. So as we were saying before, if you identify this linkage region, then you might want to go in, take the candidate genes out, and look specifically at those candidate genes. So it's kind of a multi-staged type of design. Okay, so before we can, now that we've got the basic context for linkage analysis, let's think a little more practically about what it is we need to actually begin the analysis. So we'll assume that we've decided which approach we're taking, genome-wide scan or candidate gene. Regardless of which approach we take, we still need some basic pieces of information. As I said, we're now moving into using family data. And when I talk about family data here, it can be something as simple as just pairs of related individual, like sibling pairs. Frequently, though, they are larger extended pedigrees or kindreds, okay, where we've got multiple nuclear families linked together through some upper generation. Okay. As I said, we also have to have accurate pedigree information, which we've already spent a lot of time talking about. And then in terms of the data files themselves, we talked briefly about linkage format, so the data needs to be put into linkage format. And here I just point out that you need both phenotype and genotype data. So this is actually the first time anything that we've talked about, we're now actually using genotype data. Okay, well here's information on genetic markers. I knew I'd get there eventually, but we've kind of covered this already. So genetic markers, as I said, are just simply locations in the genome where we know there's variation. If it's something like a microsatellite, for example, CA repeats or variable number of tandem repeats, VNTRs, there are many more forms of these at a particular location. That's what we call polymorphic. Versus something like SNPs, which are single nucleotide polymorphisms, there are just two alternative forms or two alleles at that location. So they're not very polymorphic, but they are much more common. Okay. And the other key thing about these genetic markers is, for the most part, they have no function in terms of thinking that they're necessarily involved with the disease, especially for SNPs. We pick these SNPs not because they have some function, at least that we know of, in relationship to our disease. We pick them because we know where they are, and they serve as an address or a signpost on the chromosome. Okay, not that they're functional. Now, there are times when you want to pick a functional variant, but that's really a different situation. Okay, so this is a lot of information here. So now when we get down to actually thinking about doing the analysis, we have, I try to break this up into kind of simple steps, and in some ways this is oversimplifying, but it'll give you the idea. That we have two different broad statistical approaches that we can take to analyzing the data. The first is what I'll call the parametric, also sometimes referred to as the model-based or the LOD score-based linkage analysis. Okay. Probably many of you have come across LOD scores in the literature, and so that might be a term that you're familiar with. We'll describe that general approach. Under each of these two broad categories, there are many different extensions and many different variations of what's considered a LOD-based method versus a non-parametric or model-free. But I'm just going to try to highlight the basic concepts and describe what are probably the most common analyses that you'll see. So we can take a parametric or model-based approach, or we can take a non-parametric or model-free approach. And the best example of the non-parametric is the SIB-pair linkage analysis, which I'll go through today. So these are the broad categories, and we'll talk about each of them so that you have an understanding of what these are. Okay, so we're going to do the parametric linkage analysis first. Okay, so with the parametric or LOD-based linkage analysis, what we're doing is, again, we are building different models, and we are comparing those models using likelihood ratio tests, or the likelihood of observing the segregation patterns of these two loci. One is your marker, whose location is known. The other is your putative susceptibility gene that you're trying to identify and localize to a particular position. In the simplest case, what we're doing is comparing two different models under two different hypotheses. Okay? Under the null hypothesis, which says there is no linkage, so in other words, there is no linkage between our marker and our susceptibility gene. In that situation, theta, which is the recombination fraction, is 1/2. And that goes back to Mendel's law of independent assortment, where genes assort independently so that you've got a 50% chance of getting one versus the other. Okay? So here we're saying they're not linked. These things assort independently. That's where the recombination fraction equals 1/2. That's our null hypothesis. The alternative hypothesis, which is what we take as evidence for linkage, is when theta is equal to anything less than 1/2. Okay? And what that says is these things are not assorting independently. They seem to be traveling together. Where it gets interesting is looking at how close this relationship is. In other words, how often or how close are these things together? Is the recombination fraction between them just slightly less than 1/2, or is it like 0, where there's no recombination? In that situation, I mean, that's an extreme situation, but it might be that that actually is the functional marker that is co-segregating. It is the disease-causing gene. What we're saying when the recombination fraction is very small, that these two things are very, very close together. That's what we like. Okay. Generally, we're in a situation where theta is not 0, but it's less than 1/2. And that tells us that, well, there's evidence for linkage here, but they're not real close together. Okay. So what we're doing is just simply, in a very simplified way, is comparing the likelihood of a model where theta is equal to 1/2 to a model where theta is some value less than 1/2. And traditionally what we do is we actually vary the value of theta and test these different models to see what model gives us the best evidence for linkage. Okay. Okay, so when we do a parametric linkage analysis or a LOD score-based linkage analysis, what this type of approach requires is that we actually specify what the underlying genetic model is. In other words, do we think that our susceptibility gene, whose location we don't know, do we think this is inherited in a dominant recessive or codominant fashion? Also, what do we think the allele frequency is of this putative susceptibility gene? We also need to know the penetrance if we're talking about a qualitative trait. We haven't really talked about penetrance. And also the transmission probabilities. Segregation analysis is where you're going to get this information. Okay? So you have to know what the underlying genetic model is. That was kind of our point in doing segregation analysis, was to figure out what's the best fitting model and what's the mode of inheritance. And then once you have that best fitting model, what was your best estimate for your allele frequencies, your transmission probabilities, etc. So for a parametric linkage analysis, you actually have to specify an underlying genetic model. The problem is, to the extent that you misspecify any of these parameters as well as the genetic model, you're going to lose power. Okay? The advantage is that LOD-based linkage analysis is one of the most powerful approaches when you have a correctly specified model. So that's kind of the trade-off here. That, depending on how well your segregation analysis went and how accurate your estimates are, you're going to have better power the more accurate those are. Okay, so a LOD score. What exactly is a LOD score? Okay, a LOD score is log to the base 10 of the odds of linkage versus the odds against linkage. So this is our theta of 1/2 versus our theta of less than 1/2. Okay? So a LOD score, don't think of it like an odds ratio. It's not exactly the same, although it's kind of what people think about when we say the log odds of linkage versus no linkage. Okay? The important things to keep in mind with a LOD score is that in general, the higher the LOD score, the better. We've tried to define some critical values for LOD scores that will help us interpret the results. And generally speaking, a LOD score of greater than 3 is considered evidence in support of linkage. This is a little bit interesting because a LOD score of 3 is really log to the base 10, so it's 10 minus 0.003, so 3 decimal places, which you would think would be highly statistically significant. But in fact, on a genome-wide scale, because we're testing so many markers, that really is equivalent to a p-value of 0.05. Okay? So this is considered minimal evidence for linkage, but it's what we tend to go with. Now, this is what a significant LOD score is. There's a lot of debate about, especially as we're going to more genetic markers now. We're testing multiple hypotheses. We're not just testing a recombination fraction of one value, but we're testing several different recombination values. So there's a lot of discussion in the literature about what an appropriate LOD score is. But generally, use LOD score of 3 as kind of your rough guide as to whether or not something is statistically significant in the positive direction. The other thing to consider is a LOD score of -2 is usually considered significant evidence against linkage. And that is probably most appropriately used when you're looking at a particular candidate gene or region. Because what you're trying to say is, we are trying to exclude this region or this gene. We're trying to show that this is not the one that we're looking for. You have to be a little bit careful here when you're excluding a region or a gene to be sure that the families that you're using are homogeneous. Because what you may inadvertently do is if a subset of the families don't show evidence for linkage, and they're the majority of the families in your sample, you could exclude an important region that actually is important in a subset of families. So if you're doing exclusion mapping, it's really important to actually look at the per family LOD score and make sure that you don't have a subset of families that actually do show evidence for linkage before you exclude this region. Also need to make sure you've got enough power to pick it up if it were there. Okay, so be a little careful with exclusion mapping, but it is used, especially, as I said, in candidate genes and excluding particular regions. The other thing about LOD scores is when you do these analyses, you get a LOD score for each family in your sample. And assuming that the families are independent, meaning that they're not somehow related to each other through marriages or whatever, then what you do is you add up the LOD score from each family to get the overall LOD score for your sample. Okay? Now, this raises a couple of issues. It's tempting when you look at these per family LOD scores, what you're going to see is the LOD scores range generally from a negative value up to positive values. Some families are much more informative. They have higher LOD scores. Other families, they actually have negative LOD scores. And when you start adding all these things together, obviously the families with the negative LOD scores bring your overall LOD score down. You can't go in post hoc and only select the families with the positive LOD scores and then add them up and publish it and have this whopping LOD score. You cannot do that unless you have a priori some reason to believe that a subset of your families are different. And if you have an a priori hypothesis, you should test that in the beginning. But you can't just go in, take the positive LOD score families, add them up, and publish it, and not mention the others. Okay? So it's kind of a mixed bag. The advantage of that is that if you, let's say, have a relatively small sample, you do your linkage analysis, and you get a LOD score of, let's say, 1.5 to 1.2. We get excited with these days. You then might be tempted to say, well, you know, if we collect some more families, we could increase our evidence for linkage, which is true. If you collect your families in the same way as your original set, you can add your LOD scores together. The danger is your next 5 families could all have a negative LOD score and bring that value down. It could also go up. It's kind of a chance you take, but it's acceptable if they've been collected in the same way under the same criteria to add those families to your LOD score. Okay? Yeah.
In analyses of randomized controlled trials, periodically data safety monitoring committees will take peeks at data, which makes you have to redefine the p-value used to reject the null hypothesis. And so it seems to me that you're peaking also, and do you have to readjust your LOD score criteria as a result?
And this is where the literature, there is a lot of debate about what an appropriate LOD score is. And this LOD score of 3, as I said, is really a rough guide, and there is a lot of information out there about what is an appropriate LOD score. There's a lot of debate. Some people are, leaning on the side of saying, well, we don't want to miss something if it's really there. And so we don't want to make these criteria too stringent that we just, ignore something or miss it. So what some people tend to feel is that use your LOD score of 3, or even in some cases less than that, and then the important thing is to replicate and follow up, okay? Because you don't want to miss it in the first place. So there, as I said, there's a lot of debate about how you adjust these LOD scores, what is a significant LOD score, what's not. And I would say with most things we do, just be cautious in your interpretation, okay? If you've got a LOD score of 3, that is probably minimally significant. Frequently, we follow up on LOD scores of less than, 2, and sometimes less than that. But the idea is that you follow up and try to verify that there's something in there. So in terms of the assumptions we're making with these parametric or LOD-based linkage analyses, is that the gene that we're looking for is inherited in a Mendelian fashion, and that there actually is a gene, that your trait is genetically influenced. With some known mode of inheritance, we know these parameter estimates. We assume that our families are homogeneous in terms of the underlying genetic reason for the disease. That the underlying genetic model is not purely polygenic, that there actually is a major gene that we can detect. We also assume no interaction between the susceptibility gene and the marker. We assume that there's random mating in our population, our pedigree data is accurate, and our pedigrees are independent, unrelated pedigrees, so that we can add those LOD scores together. A couple of things to think about that are going to affect your linkage analysis is, again, misclassification of your genotype data as well as your trait data is going to result in a loss of power. Misspecification of the genetic model also results in a loss of power. Pedigree errors. Your 3-generation pedigrees are more informative than just 2-generation pedigrees or a single-generation pedigree. Okay, that concludes the parametric linkage analysis portion for today.